Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 48. The topic is the citric acid cycle. Discussing agenda for the topic will be conversion of pyruvic acid into acetyl coenzyme A, and then we will study the citric acid cycle, which is also known as tricarboxylic acid cycle or Krebs cycle. Lecture outcome: after watching and listening this lecture, students will be able to know about how pyruvate is converted into acetyl coenzyme A and subsequent events. Introduction. As you know that pyruvate is the end product of glycolysis, so what will happen to pyruvate? What will be the fate of pyruvate? If oxygen is available, then pyruvate will enter into one pathway. If oxygen is not available, then pyruvate will enter into another pathway. So in this lecture we will basically discuss the fate of pyruvate in the presence of oxygen. After pyruvate is oxidized, the citric acid cycle completes the energy yielding oxidation of organic molecules. Glycolysis releases less than a quarter of the chemical energy in glucose that can be harvested by cells. Most of the energy remains stockpiled in the two molecules of pyruvate. So remember, glucose was a 6-carbon molecule, and the end product of glycolysis is 2 pyruvate molecules, each 3 carbon. So now when oxygen is present, the pyruvate in eukaryotic cells enters a mitochondrion where the oxidation of glucose is completed. In aerobically respiring prokaryotic cells, this process occurs in the cytosol because prokaryotes lack mitochondria, so the process occurs in the cytosol and in the plasma membrane. And later on in this chapter we will discuss the fate of pyruvate when oxygen is not available, or in a prokaryote that is unable to use oxygen, for example the anaerobes. Now the pyruvate molecule, which is the end product of glycolysis, must be converted into another molecule called acetyl coenzyme A, and then acetyl coenzyme A will enter into Krebs cycle. So the conversion of pyruvate to acetyl coenzyme A, this is a 3-step process in which 3 steps are involved, and all those steps are catalyzed by a multi-enzyme complex called pyruvate dehydrogenase. So here we will analyze these 3 steps one by one. The first step is the removal of the carboxyl group. So pyruvate is a 3-carbon molecule, and one of the carbon is released as carbon dioxide, so the remaining 2 carbon fragment is left. So the first reaction is the removal of carboxyl group in the form of carbon dioxide. The second step is the remaining 2 carbon fragment is oxidized and the electrons transfer to Nadh, storing energy in the form of NADH. So remember, 2 NADH molecules were produced in glycolysis, and then glycolysis resulted in 2 pyruvate molecules. So now each of these 2 pyruvate molecules will convert into acetyl coenzyme A, and then acetyl coenzyme A will enter into Krebs cycle. So here we are analyzing one pyruvate molecule, but remember that for each glucose molecule 2 pyruvates are produced, and then these 2 pyruvates are converted into acetyl coenzyme A, and then the 2 acetyl coenzyme A's enters into Krebs cycle. So remember those things while calculating the total number of NADH and FADH2 produced. Now in the third step, coenzyme A, a sulfur-containing compound derived from a B vitamin, is attached via its sulfur atom to the 2-carbon intermediate. So, in the third step, coenzyme A is attached with the 2-carbon fragment and it forms acetyl coenzyme A. And acetyl coenzyme A is, of course, a molecule with high potential energy, and then this molecule will enter into Krebs cycle or the citric acid cycle. So what is citric acid cycle? The citric acid cycle functions as a metabolic furnace that further oxidizes organic fuel derived from pyruvate. So at the completion of the citric acid cycle, the complete oxidation of the glucose molecule takes place. Figure 2 summarizes the inputs and outputs as pyruvate is broken down to 3 carbon dioxide molecules. How 3 carbon dioxide molecules are produced? Remember, one carbon of pyruvate is liberated in the first step when pyruvate converts to acetyl coenzyme A, and then 2 further carbon molecules are liberated as carbon dioxide in citric acid cycle. And a single citric acid cycle, or when we are considering one pyruvate molecule, so it produces 1 ATP per turn. And remember, this 1 ATP is produced through substrate level phosphorylation. The 4 ATPs which were produced in glycolysis were also produced through substrate level phosphorylation. In substrate level phosphorylation, one of the intermediates of a pathway provide phosphate group to ADP and forms ATP, and along with the NADH, FADH2 molecules are also produced. So for each pyruvate molecule entering into Krebs cycle, there will be production of 3 NADH and 1 FADH2. But remember that 1 NADH is also produced when pyruvate changes into acetyl coenzyme A. Now these are the high energy electron carriers and they will release their energy and initiate the process of electron transport chain later, where the major bulk of ATPs will be produced. The citric acid cycle is also known as tricarboxylic acid cycle or the Krebs cycle. So why it is known as Krebs cycle? Because a person named Hans Krebs elucidated this cycle in 1930. So now look at citric acid cycle in more detail. The cycle has 8 enzyme catalyzed reactions, and in step 3 and 4 the two carbons which are left in acetyl coenzyme A are also liberated as carbon dioxide in step 3 and 4. Now the acetyl group of acetyl coenzyme A joins the cycle by combining with the compound oxaloacetate. Here the acetyl coenzyme A will combine with oxaloacetate, and this oxaloacetate is reproduced or regenerated in the 8th step. That's why this process is a cycle. And citrate is the ionized form of citric acid, for which the cycle is named. This cycle is also known as citric acid cycle because of this reason. The next 7 steps decompose the citrate back to oxaloacetate. So oxaloacetate was the starting point, and it is also regenerated at the end, so that's why this process is a cycle. It is this regeneration of oxaloacetate that makes the process a cycle. Now remember, once again I am repeating that we are discussing the complete breakdown of a single glucose molecule in the presence of oxygen in a eukaryotic cell. So for a single pyruvate, when converted into acetyl coenzyme A and then entering the Krebs cycle, produces 3 NADH in step 3, 4 and 8. In step 6, FADH2 is produced instead of NADH. And then in step 5, 1 GTP is produced, and that also through substrate level phosphorylation, when one of the substrate provides phosphate group to ADP. Here instead of ATP, GTP is produced, which is also similar to ATP and performs the same functions. So now this was the structure I draw to students a year ago, and here you can analyze the citric acid cycle or the Krebs cycle or the tricarboxylic acid cycle. Remember that pyruvate was converted into acetyl coenzyme A by in a 3-step process. The process was catalyzed by pyruvate dehydrogenase enzyme, and in chemical terms you can name those 3 events into a single term, which is called oxidative decarboxylation. So acetyl coenzyme A is produced. Now this acetyl coenzyme A is a 2-carbon molecule. It combines with a 4-carbon oxaloacetate and it forms a 6-carbon molecule. And then in step 2, first water is removed and then water is added and isocitrate is formed, which is also a 6-carbon molecule. Now this is the step 3, where the first NADH is produced and the first carbon dioxide is liberated. Remember, 1 carbon dioxide also liberated in conversion of pyruvate to acetyl coenzyme A. So this is the second carbon, the first for this cycle and the second overall. And in the 4th step, NAD is converted into NADH, so this is the second NADH produced, and another carbon dioxide is also liberated. So now the initial glucose molecule has lost all its carbon molecules, 1 was liberated in conversion to acetyl coenzyme A, and 2 are liberated here, first in the third step and second in the fourth step. And this is the phase where 1 GTP is produced, the fifth phase, and this GTP is produced through substrate level phosphorylation. And then in step 6, FADH2 is produced instead of FAD. In step 8, yet another NADH is produced. So there are a total of 4 oxidation events. 4 oxidation, the first of which is occurring here, second here, third here, and fourth here. So for one pyruvate molecule entering into Krebs cycle, 1 NADH produced through conversion into acetyl coenzyme A, 3 NADH produced in Krebs cycle, and 1 FADH2 produced along with 1 ATP. So now this is once again a repetition of what we studied. So this is the Krebs cycle first, the pyruvate, is converted into acetyl coenzyme A, and in this process 1 NADH is produced. So this is for one pyruvate molecule, 1 NADH produced in conversion. This is the second NADH. This is the third NADH. This is the fourth NADH. And this is the single FADH. And this is the single ATP produced. This is for one pyruvate molecule. Remember, there are 2 pyruvate molecules. So you will have to multiply all this with 2. So 2 NADH here and 6 NADH here and 2 FADH2 here along with 2 ATPs. So now we break up the total NADH and FADH2 produced till this point. So 2 NADH were produced in glycolysis, 1 NADH was produced in conversion of 1 acetyl, 1 pyruvate into acetyl coenzyme A, and then in the Krebs cycle 3 NADH were produced. And 1 FADH2 was produced. One NADH. So I'm repeating it again. This is the NADH production of glycolysis, where 2 NADH are produced. The end product of glycolysis is 2 pyruvate molecules. So each pyruvate molecule is converted into acetyl coenzyme A, and then acetyl coenzyme A enters into Krebs cycle. So for 1 pyruvate molecule, 4 NADH and 1 FADH2 is produced. And for 2 pyruvate molecules, 8 NADH and 2 FADH2 is produced. And along with this, 4 ATPs are also produced, 2 in glycolysis and then 2 in the 2 Krebs cycles. So up till now, what is the net production of this these three steps. The net production is production of production of 10 NADH and 2 FADH2 along with 6 ATP produced. But remember, those 6 ATP are produced through substrate level phosphorylation, and these are high energy electron carriers, and they will enter into Krebs cycle, into electron transport chain, and start this process, so which we will discuss in next lecture. Thank you very much. This is the end of the lecture. Allah Hafiz.